Hello and welcome back to the classes of Mastering Biology. In the last class, we have already discussed the structure of nephron. All right. So today we shall be talking about how does a nephron help in the filtration of blood and how does it help in the formation of urine. Now, let's have a look here. When we say that the blood enters through the afferent arteriole, the filtration takes place in the glomerulus, which was a tuft of capillaries, that is held inside the Bowman's capsule, which is a cup-shaped structure. And here the filtrate gets collected, which is called as the nephric filtrate, right? Now, this filtrate, as it passes into the tubular part, which we call as the PCT, the proximal convoluted tubule, since everything has been filtered out except for some large protein, that means the useful materials have also been filtered out. So, do we release those useful materials in the form of waste? No. They have to be reabsorbed. And that reabsorption takes place here in this tubular part of nephron, which is known as the PCT. All right. What all things from the filtrate are reabsorbed, I'll just tell you in a while when we write the steps individually. Then this filtrate passes down into this U-shaped tube, which we called as the loop of Henle. And here reabsorption of water, maximum water is reabsorbed here. That means which renal tubule is helping in reabsorption? That is the PCT and the renal tubules. Then the filtrate further proceeds here into the last part, which is called as the DCT, the distal convoluted tubule. This part does not help in reabsorption, but it plays a very important role because the waste which are left out in the surrounding tissue can be directly added here in the filtrate. So this is an additional way of getting rid of the waste. It then opens into the collecting tubule and finally into the common collecting duct. These are collecting tubule from several nephrons. All right. So finally we have a collecting duct which opens to outside of the kidney into ureters. And when this filtrate is going to leave the kidneys and enter the ureters, then only we call it as urine. So I hope the basic idea of the physiology is clear. How urine formation will take place, or the question can be, what is the physiology of urine formation? How does a nephron works? Or explain in brief the functioning of the nephron. So you have to write about the nephron, how filtration takes place, how reabsorption takes place, and how urine is produced. So, my dear children, urine formation or the working of nephron it involves 3 steps. The first one is ultrafiltration. Now, why ultrafiltration? Filtration, you know, because the blood is being filtered here. But why is this word ultra added? The word ultra is added because the filtration takes place under a certain osmotic pressure. There is certain pressure. And when the filtration takes place under high pressure, it is called as ultrafiltration. All right. So, filtration of blood takes place here. Ultrafiltration is filtration of blood under pressure. All right. And where does it take place? It occurs in glomerulus. All right. All the components of blood are filtered except the large proteins. So they remain in the blood and they are passed out from the glomerulus. All right, here, this way. Now, the filtrate gets collected in the Bowman's capsule. In the Bowman's capsule. Now the filtrate is collected here in the Bowman's capsule. All right. Now, as I told you, the filtrate will now be proceeding into which part? The tubular part of the nephron. So here, the second step takes place, which is selective reabsorption. What is selective reabsorption? The nephric filtrate comprises of both useful and non-useful substances. So it has both the useful as well as the non-useful substances. Now tell me which should be reabsorbed. But obvious, why we would require something which is not useful to the body. So absorption of only the useful substance will take place, and so we say reabsorption, because once they were already absorbed and they were in the body, in the blood. So reabsorption of useful substances like glucose, amino acids, salt, and water. Takes place. Mainly these substances are reabsorbed. The reabsorption occurs in the proximal convoluted tubule and loop of Henle, or the Henle's loop. All right. In case of PCT, free surface has finger-like projections called microvilli. And what these microvilli will do? These microvilli increase the surface area for absorption. Clear? So microvilli are there to increase the surface area of absorption. And what is being absorbed? As I've already told you, reabsorption of useful substances are going to take place in the PCT and the loop of Henle. In case of loop of Henle, mainly water is reabsorbed. Mainly water is reabsorbed, but you don't need to be that precise. So you can write in a combined statement. All right. As we move to the third step, which is the third step? The third step I told you was tubular secretion. Tubular secretion occurs in DCT. This is the additional way of getting rid of waste from the body. So the surrounding tissues which have waste that were initially not present in the blood, they are directly added here in the DCT, and the filtrate enters into the collecting tubule. The collecting tubule of many nephrons open into a common collecting duct. All right. So I've already told you that the DCT will lead into a narrow collecting tubule and then finally into the collecting duct. This collecting duct will now let the filtrate leave the kidneys. And the moment it leaves the kidneys and enter into the ureters, it is now called as urine. So we can write the last point about this. The filtrate now leaves the kidneys and enter into ureters. It is now called as urine. This is now called as urine. So this is how the formation of urine takes place. These are the three steps of the urine formation. How urine is formed. First step is ultrafiltration. Second step is selective reabsorption. And the third step is the tubular secretion. I hope I'm clear with the physiology of urine formation, how urine formation takes place. All right. Now I will discuss you with a small topic, which is what is urine. So if I talk about urine, what is urine? Urine is a yellow colored fluid. The pH of urine is slightly acidic, something about 6.8. It comprises of the main composition is mainly water. It is 92% water. Then there are organic and inorganic salts, where all the metabolic wastes are also present, including the nitrogenous waste in the form of urea. All right. Besides urea, some uric acid is also released. But since urea is the main nitrogenous waste in case of human beings, so we call ourselves as ureotelic. This I have already discussed in the first lecture of excretion. If you have missed it out, please go back and revise it, and then you can continue from here. Now, the yellow colored fluid is yellow because of the presence of urochrome. This urochrome is a pigment. This urochrome is a pigment which is yellow in color, and this is what imparts yellow color to the urine. All right. So this is an overview of what is urine and what is the composition you should be knowing. Now I am left with the last part, which is dialysis or hemodialysis. So we will take up quickly in the next video.